Hello students, we have seen about cell division. This can configure in three types, like the first one is called amitosis, and we have discussed about the amitosis process. Now today we will discuss about mitosis. If you break the word, you will get two words, mitos plus osis. Mitos means equal or with thread, because here the chromosome are going to participate in the division process. That's why this is called with thread. Here the threads are chromatid thread or chromatids, while osis means stage. So it's a stage or a process where chromosome will divide, and here the parental cell, that means a 2N cell, will divide, and after the division two daughter cells are formed that contain genetic material like the mother cell. That means this one contain 2N kind of nuclei. Same here, the another daughter cell also contain 2N kind of nuclei, but the cytoplasm that will divide again in the equal manner, but this daughter cell contains half of the nuclei, half of the cytoplasm of mother cell. So so this mitosis comprises two phases. The first one is karyokinesis, that means division of nuclei, and the second stage is called cytokinesis, that means division of cytoplasm. The next point is about mitosis. This one is first discovered by Strasburger in plant cell, while this one is discovered by Fleming in animal cell. So two discoverers are here. The first one is Strasburger in plant cell, while the second one is Fleming in animal cell. But if the question asks to you the overall view about the discovery of mitosis, then you will give the preference to Fleming, because this term is also provided by Fleming. Now various names are given to mitosis on the basis of chromosomal distribution, and these names are, this one is called indirect cell division, somatic cell division, or equational cell division, because here the chromosome are going to divide equally. So first a brief review about the mitosis, later on we will see the karyokinesis stage. We have seen about the word meaning of mitos and osis. Next is about discovery of mitosis. The German biologist Strasburger in plants, while the German biologist again Fleming in animal cells. Mitosis is the cell division that takes place in somatic as well as in germinal cell. Means our somatic cells, the cells which are present in different body organ, and the second one, germinal cells, reproductive cells like gametes. So mitosis process takes place in somatic as well as in reproductive cells or germinal cells. And we have discussed that it comprises two stages. The first one is karyokinesis, that means the division of chromosome or nuclei, and the second one is cytokinesis, that was a division of cytoplasm. Now the karyokinesis stage, karyon plus kinesis, the karyon means nuclear, kinesis means division. So it's nuclear division basically, which comprises four substages, which are prophase, metaphase, anaphase, telophase. Now right now we will going to start the first stage, that's called prophase. So the first phase of mitosis, first division phase of mitosis is prophase. Again you can break the word, you will get two words. First one is pro, that means earlier, while the second mean is phase, that means a state. So it's a state where the earlier kind of process are going on. The main content of prophase is, this one is the largest division phase. I repeat, this one is the largest division phase of cell division, while the largest cell cycle phase is interphase, because that takes near about 30 hours, while the division can complete within an hour. Next, the main content of prophase is, here the chromatin thread which is present in a nucleus. First of all, that get segregated in several threads, and these threads are called chromatids. Now, these chromatids join at a point which is called now centromere, and later on they get dehydrated. They will lose the water molecule from their terminal end, and this is called the condensation of chromosome. And now, because of the losing the water, they get fold or joined together. These threads get joined together and a chromosome will form. How they will lose the water? I can simply clarify it to you. This terminal end of the thread will lose H plus ion, while the another will lose OH minus ion. So this is a water molecule here, this one. Whenever this terminal portion loses H plus, this will get negative charge. Same here, whenever this one loses OH negative, that will get a positive charge. And here the negative and positive terminal ends attract to each other. That's why they will get connected together. Same here, this terminal end will lose its H plus contained and get the negative charge, and this will lose OH negative and can get positive charge. At the last, both the arms join together and will form a chromosome. Thus a chromosome formation occurs here. This process is called condensation of chromosome. Along with this process, the nuclear envelope that get disappear, and same here, the nucleolus which is present in a nuclei that get disappear. At the last of prophase, you can see the centriole which is present nearer the nuclei, it gets separate out, and this daughter centriole will move towards the different pole. So at the last of prophase, you will get the cell like this. Here the chromosome has been formed. Next one, the nuclear envelope get disappear, or this one is going to discriminate now. Same here, the nucleolus that get break down, and the centriole will move towards the different pole. This one moves towards the north pole and another will move towards the south pole. This is the first phase, prophase. Pro means first, phases means stage, first or earlier. And the comments are regarding to prophase are, the first one, this is the largest division phase where the condensation of chromosome occur. Along with the condensation of chromosome, nuclear membrane and nucleolus get disappear. At the last of the stage, you can recognize the last or late prophase by the spindle formation. Whenever the centriole gets separated, they will move towards the terminal and they will form the spindle here. So we can configure prophase in three substages again. The early prophase where the condensation of chromosome is going on, the mid prophase where nuclear membrane and nucleolus get disappear, and the last one, late prophase, where spindle formation and movement of centriole towards the different poles. The next substage is called metaphase. The next substage is metaphase. Again, if you break the word, you will get two words, the meta word, and the second one is phase. Phase means stage, while meta means in the mid or after. So this is the phase which occur after prophase. That's why this is called metaphase. And this is the phase where chromosomes are going to arrange in the mid of the cell, at the equator of the cell. That's why this is called metaphase. In this phase, first of all, spindle formation or astral rays, which are formed by the centriole with the help of tubulin proteins, they get attached with the chromosome, the centromere of the chromosome, and they are pushing chromosome towards the equator of cell. These are the chromosome here that get attached with the these astral rays get attached with the centromere of chromosome, and now the movement of chromosome will be done towards the mid or equator of the cell. This is the equator region. Now the first of all, the tubulin protein or the astral rays get elongated, and because of it the chromosome will move towards the terminal, they will move towards the equator of cell, just like that. And because of this movement, now the chromosomes are get connected, or they are arranged at the equator or mid position of the cell, and they are forming a specific plate-like structure. They are forming a plate-like structure which is called metaphase plate. Here, these are clear. This one. Now this plate is called metaphase plate. So right now we can say the metaphase process can be configured in two parts. In the earlier metaphase or pro-metaphase, where the chromosome are going to move towards the equator, this is early metaphase. While this stage where the metaphase plate has been formed, this is called late metaphase. This is the stage where chromosomes are quite clear, they are quite shortened, thick and clear. So this is the best stage to study about the chromosome structure, shape, size, etc. So the main content of metaphase are